Thank you very much. Let's play with the technology here for a second. Well, kia ora koutou. Thanks very much, Rachel, for the very kind introduction, and thank you everyone for giving up your Saturday night. It's fantastic to see friends here, former patients, families, people who've lost people to cancer here tonight. Thank you all for coming along to hear us talk about something which is very dear to our heart and very dear to the lives of so many New Zealanders. I'm going to zoom out from Parry's molecular cell to something equally as complex, which is political systems. And equally mutable, I think. I'm going to talk about melanoma. Why am I going to talk about melanoma? Well, as many people here know, New Zealand is the melanoma capital of the world. We like beating the Aussies in many things. We've managed to beat the Aussies off their title of being world leaders in melanoma, and now we're number one. We're a country which is predominantly coastal, and we spend a lot of time in the sun. This is, of course, the beautiful St Clair Beach, just a stone's throw from my house. And sun exposure, the number one cause of melanoma, is in fact seen to be desirable. So many other carcinogens we've done a great job of beating down and saying avoid that cigarette, maybe have one less beer at dinner time. Perhaps we might do some more exercise. But as far as sun goes, we're doing a good job with our kids, with our teenagers, we're terrible. And the number of people I've met who have had cancer or melanoma who are now in their 70s and 80s, they say to me, Chris, you know, when I was a youngster, we used to put on baby oil and go and bake. Yeah, very, very common. We didn't know the ills that sun exposure would cause. And this is the sort of thing that we see. Intense burning, and that seemed to be cool because you could have a suntan when you come back from your holiday just to show your friends that you've had a great holiday. And those of us who perhaps might forget to put sunblock on our feet and yet still wear our national uniform, the jandals, as we wander around the streets. And in fact, we're not taking on the SunSmart message perhaps as well as we might think. This is just around the corner at Lake Hayes Estate, a playground where I take my kids when we go and see my brother-in-law who lives there. And you'll notice in central Otago heat a distinct lack of shade on a kid's playground. We send our kids out at morning tea time, at lunchtime, at afternoon tea, after school, for much, much more than the recommended time. And that is why we get so many of these. This is in fact the most common form of melanoma. Cutaneous melanoma, recognisable by direct dermoscopy, which is looking at this thing underneath the microscope with a bit of gel. You can see the irregular borders, the pigmentation, the discoloration, the fact they can increase in size and spread, and if they bleed or ulcerate, it's time to get one of them whipped off for sure. Very, very common in New Zealand. More than 3,000 melanomas every year are found, and more than 300 people die each year of advanced melanoma, which is more than the road toll. And there's a lot we can do to prevent it. Now, when I was doing my specialist training as a registrar, we had literally no meaningful treatments at all for melanoma. And one of the first patients I ever met with advanced melanoma was this chap, Kyle. He was a guy from just over the road at Wanaka. He was 35, had 2 young kids. He also liked playing backyard cricket with his kids. He grew up in a cricket field pretty much. Down there with his younger brother throwing a ball around, taking turns batting and bowling. He ran a pub and he found a wee spot in his leg, had it cut out, came back as a melanoma, didn't think too much more about it until he started noticing a number of lumps that were popping up under his skin. And he went along to his GP and he had a needle aspiration test done where they take some cells out of the lump. They looked at it under the microscope and the classic pigmented melanoma cells came back from those lumps. So we got it cut off, but more came back. And he got more cut off and more came back. So he was referred to our service and we talked with him about melanoma, the fact that the cancer had spread to multiple parts of the skin, not to other parts of the body at the time. And we had nothing, really. So he headed off to Australia, the Melanoma Institute of Australia, and he had a bit of the tumour mashed up into a bowl and turned into a vaccine with their technology there, and had the vaccine treatment. But still the cancer came back, and he had a number of these spots continuously cut off. At one stage he was getting 1 or 2 a week that were popping up underneath the skin. And while it was in the skin, although it was terrifying for him to have this number of new spots turning up, we could kind of keep up with it by catching up and having multiple surgical attempts at that. But then the melanoma went from the skin and it started going inside, and it spread to his inside lymph glands, into his liver, and then it did this. This is a CT scan of his brain. And those white haloed round dots are brain secondaries. So this was a 36-year-old chap, 2 young kids, previously an independent businessman, now confined to a wheelchair. He had to have his wife wipe his bum for him, and his brother helped him shower. And that is, he succumbed to his melanoma from that, and that was the last few weeks of his life. We had nothing to offer this guy. It felt like we were watching a train moving slowly along the tracks, desperate to try anything that we could to try and slow it down. And the reality was we had nothing. That all changed in about 2010. Now, I remember this very well because my wife was very heavily pregnant at the time with our youngest child, who's now 7. And in 2010, we had the very first agent that has ever been shown to improve overall survival in advanced melanoma, Ipilimumab, or Yervoy as it's known by its trade name. Now, this is one of the immune therapy drugs, the first generation immune therapy drugs that Perry alluded to earlier on. And what this drug does is it sort of amps up the immune system, and it creates more T cells to go out and become active and swirl around and try and gobble up the cancer cells. So we previously thought it might have been a recognition error, but perhaps part of it was a numbers game, not enough immune cells. And by activating more immune cells, we could in fact get some of these T cells into the tumours, and actually finally, after 35 years of research, after trying every single known chemotherapy drug without a meaningful response, we finally found a drug that started to work in some people. This is a brief précis of the study. What the study did was it took patients and it divided them into 3 groups: patients with an old vaccine type adjuvant, and then patients in another group where they gave the vaccine adjuvant and the Ipilimumab or Yervoy. And they found that on average, the people who got the Ipilimumab or the Yervoy lived on average a number of additional months, not many months, but additional months, over the group of people who just got the adjuvant immune antigen itself. But what we found in that was that there was a small tail of people, this line out here, a small tail of people there, who in actual fact, a number of them, perhaps 20 or 30% of people who got this immune therapy drug, actually they didn't relapse. In actual fact, this small number of people who received this treatment were still alive 5 years later, and as time went on, we found they could live up to 10 years later. And so from having a disease that was almost universally fatal within 6 to 9 months, we found we were now having people who were living 5 and sometimes 10 years. A small number, but a glimmer, a glimpse of hope that perhaps would found something that might finally work against this disease. But for many, many people, it had no activity at all, and it caused a large number of side effects. The cost for 4 doses, $155,000. Mind-bogglingly expensive. But it showed that the researchers, the clever guys, were on the right track, and perhaps with a little bit more work we might get a little bit further. Now, what clinicians had known for a number of years was that melanoma wasn't just one disease. There's not just the skin cancer type that you see. We also see different types of clinical melanoma. In the top right, you see subungual or acral melanoma, which occurs underneath the nail beds. Now, the nail beds don't actually usually get that much sun exposure, and that's not really UV related. We have this other type of melanoma, which isn't that usual irregular brown pigmented, but it's more that rounded, almost blistered type, which is called a nodular subtype of melanoma. And then you've got this pigmented lesion on the inside of the person's mouth, which is called mucosal melanoma. And you can see that anywhere in the gut, even on the inside. So in actual fact, you don't have to see any sun at all on the inside of your gut, and you can still get melanoma there. So we've known for a long time that there are different types of melanoma. It's not just one disease. So it's perhaps no surprise that when geneticists like Perry got their hands on these melanoma samples, they could start to do deep sequencing and uncover the internal machinery, and they found that melanoma wasn't one disease, but melanoma in fact had a number of different molecular and genetic profiles. And as Perry had said, we could now start to target these individual mutated pathways and develop individual drugs for them and see if we could stop melanoma in its tracks. Try the immune therapy, it worked a wee bit. Let's see if we can have a go on the inside and see if we can target that. Now, all too many times up until this point, we've seen overexpressed pathways, dysregulated pathways. Let's target it and see if it works, and it doesn't. But this, indeed, when we tried it in melanoma, we saw something quite different. November 2010, my son had been alive for about a month, and we took him off to Australia to hear the results of the study that we'd participated in, the BRIM-3 study, where we tried Vemurafenib in patients with advanced melanoma. And we sat there and we watched the results coming out from this large international study, and everything changed. Now, like Perry I also like graphs. I'm an oncologist. We like data, we like facts and figures, and I'm going to take you through this. This is called a waterfall plot. Now, each of these lines, which looks okay on my screen, doesn't project quite as well on there, but each of these lines, again, represents an individual patient. A line above the graph, above the middle point, is a patient whose cancer grows on the treatment. And every line below the midpoint there is a patient whose cancer shrinks on the treatment. And what I think you can see from that graph is that almost all of the patients who started on this drug, Vemurafenib, experienced massive shrinkage in their tumour. Now, with standard chemotherapy, we might see a bit of tumour shrinkage in about a third of the patients. We might see a minor stabilisation about a third, and you see growth in the rest. But for the majority of patients, it does a small amount. We had never seen responses in oncology like this before, where 90 to 95% of the patients experienced some kind of tumour shrinkage or stabilisation on this. And it was quick. The very first patient I treated with this, again, had a lot of skin secondaries, and they stopped popping up after 48 hours. They started shrinking after 72 hours, and within a week they were almost gone, much like that slide that Perry showed. The second patient I treated with this drug came into my clinic and was in a wheelchair, and she was yellow from all of the tumour that was in her liver, that had overwhelmed her liver, and she's on her way to the hospice. And she said, well, you know, I know I'm off the way to the hospice, I've got this incurable illness, the doctor's already told me that, but how about, I thought I'd just pop by and see if there was anything you could perhaps do for me. I said, well, on your way to the hospice, just take this bottle with you of these pills and see how you go. So she headed off. About half an hour later, I got a call from the hospice doctor. What are you doing, Chris? You're giving this patient who's dying chemotherapy. What are you doing? And I said, oh, it's not quite chemo. It's just, you know, just give it a couple of days, see how it goes. Just go with me on this. And 3 days later, the pain had gone. And 5 days later, she was out of her wheelchair and she walked out of the hospice. Incredible. The hospice doctor rang me up and said, oh, we did a really good job with that lady, Chris. And she was my patient for many, many months after that and did very well for a period, but again developed drug resistance and again eventually succumbed to the disease. All of these new technologies and these innovations with the immune therapies and with the BRAF inhibitors, they still in New Zealand remained unavailable for a period after we had started doing these studies and after these results had been published. And that became pretty real for our department in the oncology unit at Dunedin Hospital when one of our own staff members developed melanoma. Alison actually was a staff member of mine, and I tell the story with the full permission of her family. Alison was a research nurse who worked in our department and in fact contributed many of the work to the studies that I just mentioned to you. Alison had a melanoma less than 1 millimetre in thickness on her back about 8 years before she was diagnosed with recurrent melanoma. 8 years on her back. She woke up one morning and said to her husband, "I think I've got a lump on my breast." And her husband said, "Yeah, well, I think I can feel that too. We should probably get that checked out." And like most women in their 40s, they would think if you've got a lump in your breast, it's a breast cancer. And so then when they got the biopsy results back, and in fact it was a melanoma, that came as a great surprise. And again, like Kyle, the first patient who I met, she started to develop 5, 10, 20 secondaries every single week. And again, then those tumours started to spread to her internal organs and the adrenal glands. Now, Alison had a BRAF activating mutation as well. And through a compassionate access program, we managed to get her onto some of these treatments. And again, with the BRAF inhibitors, her cancer stopped growing within 24 hours, started to shrink within 48 hours, and again within a week, the tumours had started to disappear. Now, she was having 20 cancers cut out every single week. 20. She would wake up every morning, and she had this diagram that she would draw, and she would feel and mark where she had new lumps every single morning. So she knew exactly where these things were. And within days, new lesions had stopped appearing and had started to shrink. So literally before our very eyes, these cancers were disappearing. Another graph. This is a graph which shows the number of people whose cancers is controlled by these drugs compared to placebo, and it is as time ticks on. And what the graph shows in summary form is that as time ticks on, more and more people unfortunately do develop resistance and their cancers progress. But even with these drugs, around about 10 or 20% of people do remain in remission with these drugs, and I still have some patients who are around 5 years later on these treatments who are still in remission. So it is indeed quite remarkable. And these were revolutionary, knowing that we'd seen Kyle dying of brain secondaries, unable to do a thing. And then we were seeing patient after patient on our studies and on our access programs were starting to respond so quickly and so profoundly. And we wanted these treatments available for all of our patients that came through, not just the lucky few who could get onto the trial or onto the access program. And it was with some trepidation and perhaps a little bit of excitement that the drugs went before PHARMAC, and we thought, can't wait. Now that we've got a drug that actually works better than anything else we've got in almost any other area of oncology, surely we're going to get our hands on these. And then PHARMAC went through and they did their review. PHARMAC said, we have noted that these drugs do work, and we have noted that there is another treatment, chemotherapy called dacarbazine, that causes a response in patients around about 1 in 6. The interesting thing about that is that that drug, dacarbazine, has never in a single study ever been shown to prolong patients' lives by a day. Not one. And because it might cause tumour shrinkage but doesn't extend life, it's hardly used at all. And it causes people's hair to fall out, it causes their immune system to drop, causes nausea and vomiting, and it causes fatigue. So it's a drug that we barely use at all, unless in extreme circumstances. So they said, we've got this drug, we're already funding something, but really the rest of us knew it didn't actually work. And they said that this new drug, Vemurafenib, hardly conferred any benefit at all, and they said, what they say, it was a high unmet need, but for most patients it was pretty modest, and it provided only a small, short-term benefit. It was small, despite the fact that the research was published in the world's leading medical journal, and it showed that we made people live longer and better with fewer side effects than we did if they had other treatment. So that was a pretty bitter pill for us to swallow. 6 months later, they reviewed the Ipilimumab, the immune therapy, and again, given the high price of the immune therapy, we were perhaps a little bit less surprised that this one didn't get across the line. They said that they were unconvinced about the long-term data, despite there being 5 years worth of data available at that time, and they felt that the side effects of the drug were too hazardous to justify the benefits. Which again is interesting, because PHARMAC's job is not to establish whether a drug is safe. That's the job of our medicines safety regulator, who had deemed that the drug was safe to be on the market and the benefits of the drug outweighed the harms. PHARMAC's job is to work out if the drug is cost effective. And we all felt that they were using weasel words in order to present the fact that actually it just cost a bit much, which actually if they come out and said that, we probably would have agreed with them. And then the next phase of cancer drugs came out, the next phase of immune therapies, the PD-1 drugs. Now, I'm a little bit simpler than our very clever scientists, and my immune biology is very much need to know. I know what I need to. And I like to think of the immune response, the PD-1, as a little bit like a rugby game. As the T cell comes a little bit closer to the cancer cell and it tries to attack the cancer cell puts out a fend and it stops the T cell from actually getting close. Now, when you give the PD-1 antibody, it takes the arm away, and so the T cell can then get in there and tackle the cancer. So there we have our ineffective T cell, and there we have our rampaging melanoma. Now, the rest of the world had moved on from the notion that chemotherapy was any kind of standard of care, and the rest of the world had accepted these new tablets, the BRAF inhibitors, and the immune therapies, Ipilimumab, were the new gold standard, and that is what everything else should be indexed against. And the new PD-1 inhibitors came out, and they compared the responses and the duration of response. These are the waterfall plots again. Compared the new PD-1 drugs to the old first-generation immune therapies. And what they found was the responses were stronger, they were deeper, and they occurred in more patients, and they had fewer side effects. So a genuine advance over the old first-generation immune therapies. Again, still up to a third of patients got no benefit, but 2/3 had at least some shrinkage and a prolongation of their life on these new drugs. So pretty exciting now, from nothing to 3 new drugs, still not a single one available to our patients in New Zealand. Now, I had some patients who were lucky enough to get access to these drugs because they had money or they had insurance policies that might cover them to a certain extent. And here, for example, was one of my patients who was on the PD-1 drug, one of my first patients on that, and he had a lung tumour. This is the lung tumour here, this great big white blob there. And after only 12 weeks on this drug, the blob had shrunk away to that small. So again, within a very, very, very short space of time, we were starting to see these tumours shrink and shrink and shrink. And I had the great pleasure of sharing that shrinkage with him live on national television as he wanted to tell his story about his access to the immune therapies and how well they'd gone for him. Now this guy, Michael, was then able to get back to farming, hauling sheep over his shoulders, earning money and being a productive member of society for a number of years after his initial diagnosis. And to look at him on the street, you'd never know that he had cancer at all. So, surely third time lucky. A third drug goes in front of PHARMAC, and this drug is cheaper, it's stronger, it lasts longer, and we get survivors. Surely we've got to get something across the line. And PHARMAC said, yeah, nah. PHARMAC said, well, maybe it's an excessively high price and the benefits are uncertain. There's a number of uncertainties in the clinical trial design. So we'll give it a low priority, which basically means they're never going to fund it. And we'll go back and we'll relook at the old drug, Ipilimumab, the first generation one, which the rest of the world has moved on. And all of us clinicians at that time felt very much like we were chasing our tails. And the message that was being shared in public with the public from PHARMAC was that the reason you don't have a melanoma drug is because they're not very good. They don't really have very good long-term data out there. We're not that convinced of the responses, and we're just a little bit uncertain about the duration of benefit. Now that was despite multiple randomized phase 3 studies occurring and being published in the world-leading medical journals. And many of us were starting to become pretty pissed off at this point with what was going on. Again, knowing that the rest of the world had moved on, and still in our country, the country with the highest rate of melanoma in the world, still had that. So in end of 2015, I was appointed as the Cancer Society's medical director, and we started to get asked a lot about these new drugs and what we thought about the lack of access to funding of these drugs. Now, it's a pretty controversial area for the Cancer Society to go into. Are we going to advocate for a single drug over our drug regulatory agency, our drug funding agency? Is this something we should do? Should we take a position on every particular drug that comes through? Well, we started to get into this a little bit because we kept getting asked all the time about what we thought about these immune therapies. And when we talked about it, we reflected on the fact that there were a lot of unanswered questions, and the fact that a lot of other countries with similar systems and similar structures like Australia, like Canada, like England, had come to vastly different decisions about these drugs than what our country had. And we said, we just don't know why they've made these decisions. We have got no ability to engage with PHARMAC. Their data's not clear, their analysis isn't clear, and the reasons why they're turning it down are just not clear. And the patients who were advocating for these were knocking on the doors of PHARMAC and were just not being heard. Now, it's fine to say a drug is too expensive. It's fine to say, actually, that costs so much, we would rather spend the money on hips, we would rather spend the money on hernias and gallbladders, we'd rather spend the money on brain cancer drugs. But that wasn't the debate we were having. And by this time, the patients, of course, in the age of the internet, were well over this. They all knew what was going on. They were in contact with people around the world when they got diagnosed with their melanoma, finding out what was the new international standard of care and why they couldn't have access to it. And two people came into my life in 2016 who I will never forget. Jeff Patterson and Lisa Renwick. Now, Jeff and Lisa, between them, organised a petition which got 43,000 signatures calling on the government to look into the funding of Keytruda. And Keytruda was a shop window. Interestingly, both Jeff and Lisa actually got a BRAF inhibitor up front. They benefited from that, but they ran the campaign for Keytruda. Jeff was 26. He had advanced melanoma, and he was 26, and he was giving his time to this campaign. Lisa was a school teacher from Tauranga who was told there's nothing more to be done, you had advanced cancer, we're going to refer to the hospice. And she found out about one of these drugs, got herself off to a private provider and got herself on the BRAF inhibitor. Jonathan Coleman, the then minister, actually refused to receive the petition, said he was too busy to meet with the patients until he was on checkpoint and John Campbell was asking him why he wouldn't meet. Then a space seemed to appear in his diary. And that was pretty offensive to the people who had gone to so much time to organise those things. And PHARMAC still kept on with their lines by saying that actually only 1/3 of people benefit from these drugs or experience tumour shrinkage, and they are a very high price, which is true. And if it's that good a drug, it would be funded. That's what they said. It's kind of a self-fulfilling prophecy at that point, don't you think? And the interesting thing again, if you look at the waterfall plots, they said only 1/3 of patients experience tumour shrinkage, but that's not true. You can see from the waterfall plots, 2/3 of the bars are below the line. That's 2/3 of the patient are getting tumour shrinkage. It's just because of the technical medical definition of what constitutes a technical bidimensional CT scanning response for the purposes of the study. But 2/3 of the patients got benefits from them. After a lot of lobbying, a lot of political pressure, a number of weeks on the headlines, and a number of very, very tough interviews of the Minister of Health, all of a sudden, 23 million extra was found for PHARMAC in their budget. PHARMAC never asked for a cent. They were told they needed to have another 23 million, and we had a Keytruda-like drug, Opdivo, was funded. Now, it's Pepsi and Coke, they're pretty similar. The clinicians had absolutely no problem with either of these drugs being funded. We're delighted that one of them got over the line. So again, the drug got funded, and then a couple of months later, Keytruda got funded as well, and what they said was that they had reached more favourable commercial terms with the company, which perhaps was the underlying reason the entire time. Interestingly, another line which they released in their decision was that as a result of evolution of the evidence, PHARMAC was more confident in the clinical efficacy of the drug. Now, interestingly, and this is a fact, between the time of the first application being declined by PHARMAC and the funding, there was not one single additional scientific paper published in the literature that updated the data or proved improved responses. Not one. Okay? Not a single paper was published in that time. And yet we had this line saying that there was additional evidence for the efficacy of the drug. And that was pretty disappointing from our regulatory agency. Do I think PHARMAC should be scrapped? No, I don't. They do a very tough job and they manage the pharmaceutical budget of 800 million and have to make prioritisation decisions based on very, very disparate conditions and a large number of people, and they manage it on our behalf. But the nagging question I could not get past was, could they ever be wrong? And if they could ever be wrong, what can you do about it? Well, the PHARMAC model is they get a fixed budget. They get told each year, they negotiate with the DHBs how much they're going to spend. That's 800 million. They then look at the evidence for new drugs, they estimate the costs, give a priority, and when the money becomes available, they'll fund within their overall budget. So they've got 800 million to spend. So let's imagine a brand new drug comes along and it might push them from 800 to 820 million, but it's great and it saves zillions of lives. It wouldn't actually get funded because it pushes them outside the envelope. They've got to stay within that. That's their model. They have advisory committees made up of various clinical experts, and they've got a cancer subcommittee made up of around 5 or 6 people. There was only one person who treated melanoma on the cancer subcommittee, and there's a dozen of us who treat melanoma around the country. The principles that PHARMAC run by, and that I happen to agree with, is that there's not enough money for everything. I think we can all agree with that. We also should all agree, I think, that actually it would be wrong for taxpayers to write a blank cheque to Big Pharma and pay those sorts of 150 grand for 4 doses prices, because then they could just charge what they want. And we must prioritise within the available money. That goes without saying. That's absolutely and utterly right. But also, PHARMAC work on our behalf. And if they are a government institution, then surely they must be accountable to the people they serve, us. And surely transparency is essential for that accountability. So we've got to know the sorts of reasons they're making the decisions that they do. And they made a couple of errors on this one. They misestimated the cost impact of the Keytruda decision by $20 million. They got their numbers wrong by 20 million bucks. And that makes me wonder if their projections were that wrong, if they perhaps misestimated the cost to go outside their envelope, and that meant people missed out for longer. But it's also fair to say, in PHARMAC's defence, if I may for a moment, is that these individual new drugs make a massive disparate impact on the cancer budget. So in fact, half of the cancer budget is accounted for by 4 drugs. Okay, half of all of our spend by 4 drugs. So you add one more like melanoma and it pushes your budget up disproportionately, and that's pretty tough. But having said all of that, I still think we need to change. We need to have a system that engages with the clinicians, that engages with the patients, that engages with the families, and engages with the community with the people that PHARMAC is supposed to serve. We need to have some kind of transparency. So instead of having this game of hide and seek where they say it's not really an effective drug when in fact it is, it's just too expensive, we do something like saying, well, let's give these guys a drug scorecard. And there are internationally validated scales that you can use, like the European Society of Medical Oncology's magnitude of benefit scale, where it gets a scale of 1 to 5 based on how good the drug is. And that would give us an idea of what kind of ballpark it is. And those questions around, is it a wonder drug, could be tested and checked. I think that we should have some thresholds for cost, whereby if it meets a certain point of effectiveness and it's at a certain price, then perhaps you should actually fund that. So if you get extra health gains, you should also pay a little bit more for that. And in actual fact, it's very reasonable to compare the benefit you get from health and from drug spend to other kinds of government spends as well. So we spend more than 2 million per life saved on roading. Do we spend the same in cancer? No, we spend 26,000 per life saved in cancer. So a lot less is spent on medicines than is spent in other areas saving lives. And I think we need some certainty. The problem as well, for many people who were waiting while PHARMAC went through their machinations, there were people whose condition deteriorated to the point by when they made a decision, they could no longer receive the drug. And I want to reflect in particular on Jeff, the 26-year-old who spent the last 6 months of his life campaigning for a new cancer drug. Jeff was on the RAF inhibitor for a while and then he became sick. He went off to have some radiotherapy and then unfortunately his cancer began to grow. Jeff and his family didn't have the money to afford Keytruda and his condition deteriorated to the point whereby he could no longer receive it, and he had to be on high doses of steroids. Jeff died on a Friday, and for a 26-year-old to spend the last 6 months of his life campaigning for a drug that he was told was no good, that he knew wasn't right, was told no particular time frame, and the minister wouldn't even meet with these guys to receive their petition of 45,000 people. I think guys like him and his family would pretty rightly feel pretty cheated. And I think if we don't do something different, we'll continue to shortchange those people. And Jeff is very much the human face of the inefficiency of bureaucracy. He is very much the face and the result and the consequence of what happens when you take a long time to make a decision. And we need to do something a little bit different to make sure we look after all the people in Jeff's situation in the future. Because if you keep doing the same thing, we're going to keep getting the same result. And that's why we need change.
So does anyone have any questions?